right, anatomy students, we're going to pick up where we left off with antigen presentation. And I believe that we were right over here where our macrophage is taking fragments of the antigen and attaching it to a major histocompatibility complex protein. And the combination of the MHC protein plus the antigen is what is going to activate our helper T cell. And now that our helper T cell is activated, he can send out cytokines to stimulate cytotoxic T cells to divide and to form more cytotoxic T cells and memory T cells. And then those cytokines can also activate B cells to start dividing to form more memory B cells and plasma cells as well. Those cytokines are also going to stimulate the B cells to produce more antigens in greater, or more antibodies, in greater quantities. And so this is another example of what's happening during antigen presentation. And so as I said, here we've got our antigen. It's being devoured by our antigen presentation cell, which in this case is a dendritic cell. It's going to take a fragment of that antigen and attach it to an MHC or self protein. And here you can see that we've got the little red antigen fragment there as well. Now, when it meets up with our helper T cell, the helper T cell receptor has to recognize both the yellow part and the red part at the same time. So it has to recognize self protein and antigen protein. And the reason for this is because antigens are tricky. They are going to try to trick helper T cells into increasing the inflammation response. Because like I said yesterday, if you have too much inflammation and too many cytokines, you're going to damage and weaken cells in the tissues. And if you've got damaged and weakened cells, then that means that they're more susceptible to infection and thus the spread of this antigen. And so the helper T cell, to try and keep all of that inflammation and the secretion of cytokines under wrap, that helper T cell needs to recognize the MHC self protein as well as the antigen at the same time. And then once that happens, our helper T cell releases cytokines, and the cytokines are going to stimulate our cytotoxic T cells to divide and form memory T cells plus more cytotoxic T cells. And it's also going to stimulate our B cells to divide and produce more plasma cells as well as more memory B cells. So those are all of the things that's going to happen when we have antigen presentation occurring here. So I just wanted to make mention that antigen presentation, while necessary for the primary response to occur, it is not necessary for the secondary response to occur, because the second time that the antigen comes around, we've already established memory, we've already established immunity, and that's one of the results that happens from antigen presentation. And so we've already gotten there, so we don't need this to happen again. This is only going to happen during our primary response, during the initial infection by an antigen. All right, so now we are going to talk about the secondary response of specific immunity. And I just wanted to mention to you that on my slides, I kind of color-coded them. So my slides that show the primary response have headings that are in green. Secondary response has headings that are in pink. So here we go. So now that we've established memory with a supply of memory T cells and memory B cells, what's going to happen when the antigen returns? Well, with a second exposure, and even with a third or a fourth or a 10th exposure, like I said, we don't need to have antigen presentation because we have memory cells already, and they are going to provide a customized attack on this particular antigen. Because we already have memory, and because our body has already learned what antibodies are needed and things like that, the secondary response is more successful than the primary response in the way that the secondary response is going to occur faster than our primary response will. And so because we already have memory, we're going to recognize this antigen very quickly, and we're going to be able to clear antigen-infected self cells very quickly as well. When our memory cells meet up with this antigen again, we are going to stimulate the production of more antibodies at a very fast rate. And again, the person is immune, so they are not going to get sick by this secondary exposure or the third time or the 10th time, because our bodies already know how to fight it. This is the rest of the picture from your textbook that I had showed you earlier on. And so up here at the top, we had the primary response, and I told you that after proliferation and differentiation, we have more memory B cells that are going to be produced. And these memory B cells are not just going to hang out in the lymph nodes, they're going to circulate around the body looking for their particular antigen to make sure that we can stop it in its tracks before it causes us to feel sick. When it encounters that antigen, we're going to have direct activation of our B cells. They're going to make a plethora of plasma cells, which are going to create lots and lots of antibodies at a very high rate. And then it's also going to create more memory B cells that are going to remain in our bloodstream. And they are going to again circulate throughout the body, making sure that we don't get infected by the same antigen again. And so I just wanted to talk about two different kinds of specific immunity. We have humoral specific immunity and cell-mediated specific immunity. And humoral immunity refers to B cells because B cells create antibodies, and any mechanism that involves antibodies in the immune response is considered humoral activity. Cell-mediated involves T cells, and again, I've color-coded it, so we'll talk about that when we see the green font on the slide. But anyway, B cells are going to produce antibodies, and you guys could take like whole classes. You could frame a whole career on the study of antibodies and how they work. But generally, they are proteins. They all have the same basic structure. The only thing that's going to be different is this tiny portion here called the variable chain, which is creating a unique antigen body site for each type of antibody. Antibodies are called immunoglobulins, and they're usually abbreviated as IG, like big I, little g, and then another capital letter. So this is called IgG or IgM. And different antibodies are going to be synthesized and secreted dependent upon the type of antigen that they need to fight. And like so, for instance, if it's an allergen, then you have a lot of IgE that is being produced. So antigens are going to bind to the surface, or antibodies are going to bind to the surface of antigens, and that's going to ease phagocytosis. It's also going to clump antigens together. So I mentioned before that antibodies can often grab onto and latch onto multiple antigens at one time and clump them all together, and that will make the clump more susceptible to phagocytosis. Remember that antibodies are going to fight against antigens that are extracellular, that are found outside of the cells. These are antigens that are flowing freely in the blood and in the lymph. They are not going to be effective against antigens that have already infected our cells. So this is a graph that you also have in your textbook, and this is just showing you the speed at which the primary and secondary responses occur and the concentrations of antibodies that are produced. And so if we start here, blammo, we are infected by an antigen that we haven't encountered yet, and you can see, look at our antibody concentration is at 0 for like the first week, and then it finally starts to pick up. Well, what's happening during this first week? Well, our innate immune response is working. We have dendritic cells that are performing antigen presentation to T cells. We have B cells that are lying in wait in the lymph nodes, waiting for this antigen to enter the lymph nodes so that it can be directly activated and then divide and form plasma cells in order to make the antibodies even. So all of that takes time, and that's why it takes about a week for antibodies to even start being produced, and then it takes another full week for the concentration of antibodies to even reach their peak. After that, the antibodies are going to start to work, and our immune system is going to start to work to decrease the amount of antigen that we have in our system. And therefore, the amount of antibody is also going to decrease because we don't need it. But look it's not going to ever reach 0 again. Here, blammo, we get exposed to the same antigen again. But look at this. It takes like a day for all of this antibody to be produced. It doesn't take as long because we have memory cells already. We already learned how to make the antibodies to fight this particular antigen. So we are able to do it not only swiftly, but in much higher concentrations. Look at how high the peak concentration is in the secondary response in comparison to our primary response. And so we're going to be able to clear this antigen from our bodies much more swiftly. So cell-mediated immunity is happening both during the primary response and during the secondary response. And cell-mediated immunity is most effective against infected self cells, cells of our own tissues that have been infected by these antigens. Our cells, our T cells, are not going to be effective against free antigens. So remember, like antibodies are going to work against free antigens, not our T cells. They're going to work against antigens that have already infected our self cells. So we have a number of different T cells that I wanted to kind of run through and explain to you. For instance, helper T cells are the ones that receive the antigen from antigen-presenting cells. And when they are presented with an antigen that matches their receptors, they're going to release cytokines. These chemicals are going to stimulate cytotoxic T cells to divide to form more cytotoxic T cells as well as memory T cells. They are going to, helper T cells are going to release cytokines to stimulate B cells to proliferate and differentiate into plasma cells to make antibodies, and into memory B cells again to just create that foundation for immunity. They're also going to stimulate B cells to create higher concentrations of antibodies at faster and faster rates. Cytotoxic T cells are the killers of the group. They are going to use chemicals to kill infected self cells so that the spread of the antigen is slowed down or stopped completely. And they're going to use the same kind of chemicals that the natural killer cells use in order to kind of dissolve the infected self cells from the inside out. But all of this is kind of kept under wraps by other kinds of T cells that are called regulatory T cells. These are going to prevent the immune system from overreacting to antigens because remember, too much cytokines especially are going to be detrimental to the healthy cells and tissues of our body. And again, if we have too much cytokine being produced, then we're going to weaken our tissues, and that is going to allow antigens to come in and proliferate and cause more infection in our body. Once B and T cells have been activated by their specific antigens, they are no longer considered just immunocompetent. Now they are mature. And they're not just going to hang out in the lymph nodes. No, they're going to circulate in the lymph and in the blood in order to kind of patrol our body to make sure that their specific antigen hasn't re-entered. And if it has, it's going to mount that specific immune response against that antigen. All right, that's it for part 2. Stay tuned for part 3.